Rose and everybody in San Carlos for the invitation to come here. It's always fun to come back to Brazil. Sorry I missed this morning because I had to go to Campinas for a thesis defense of one of my students who spent a year with me 2 years ago. And I have been informed that because of last night I have to speak very quietly because the other foreigners have now learned 3 words in Portuguese: cerveja, cachaça and ressaca. So I've been asked to speak very quietly. Okay, so as Rose said, I'm a platinum chemist and mostly dealing with inorganic chemistry and medicine, which is the subject of my mini course. And I like to start off by pointing out that really metals in biology and medicine encompasses 4 situations. One is where we have what we would call a steady state, with metalloproteins and essential metals acting in their normal role, iron, copper, zinc. But now we have much more understanding of what happens in non-steady state, where you have an excess or deficiency of a metal, copper or iron, but also a recognition, since 30% of all proteins are metalloproteins, the recognition that mutations in metalloproteins actually make metalloproteins as valid targets themselves. And it's an interesting thing to examine that aspect. Thirdly, we have toxic metals. How do they actually act? There's no such thing as a toxic metal. There's a toxic metal ion. I like to point out that mercury is not toxic. Mercury 2+ is toxic. And finally, we have medicinal chemistry, where with the right pharmacology and pharmacokinetics, we can develop small molecule therapeutics, imaging agents, and radioisotopes. And that's what I'm going to talk about today. Here are some of the major examples. Lithium carbonate, of course, we can claim as an inorganic ion because it's the lithium plus is the smallest drug. Technetium and gadolinium are imaging agents. Over 1 million doses of technetium are used in American hospitals every day for imaging tissue. If you've had an MRI, you might well have received gadolinium as a contrast agent. And the major use of metal ions is platinum in cancer chemotherapy, and that's what I'll talk about. Some small use of gold, and this value is probably maybe around $8 to $10 billion a year, which is not insubstantial. It's not as big as the $300 billion that organic chemistry would claim, but nevertheless, I wouldn't mind $10 billion. That'd be okay by me. Okay, so what I'm going to talk about is I'm going to give a brief summary of what Rose called our polynuclear platinum strategy, and then I'm going to talk about some non-covalent interactions and development of new targets. And I probably won't get to this because one of the disadvantages of speaking last is that I'm the only person keeping you from more cerveja and more cachaça, so this is always dangerous, you know. So a brief timeline for those of you who don't know. Cisplatin was discovered by accident, by serendipity, we always say. In the late 60s, the anti-cancer activity. There was a rapid development in 1978 where it entered the clinic. It's a small square planar molecule, not tetrahedral, square planar. And we absolutely no carbon, so the first genuinely inorganic therapeutic drug. We believe that the chlorides are displaced by DNA bases, and this platinum ammonia group stays intact. It is used in metastatic testicular cancer, ovarian cancer, and transitional bladder cancer. The compound had significant nephrotoxicity as well as nausea, and so the second generation compound, carboplatin, is basically general chemistry put into action. Instead of having relatively substitution labile groups like chloride, a chelating dicarboxylate was prepared, which now gets excreted intact. This does not get excreted intact. It binds to proteins in the kidney. This gets excreted as an intact molecule, and this has now become first line for ovarian cancer. The mechanism is roughly the same as you might expect, because once we hydrolyze this carboxylate, we're going to end up with the same unit. So this is now ovarian cancer first line treatment. And then there was a gap till about 2002, where you can now, instead of changing the leaving groups, you change the carrier group ammonia, and 1,2-diaminocyclohexane was used, and in the presence of another oxygen group, oxalate. This has now been approved worldwide for metastatic colorectal cancer in combination with 5-fluorouracil. So these three drugs are the mainstay of cytotoxic therapy. They're the most prescribed anti-cancer drugs in the clinic. There's a sort of a contradiction, since we're the spokespersons for metals in medicine, but it's a contradiction that no drug companies work on platinum. It's cytotoxic. It's not as popular, shall we say, as targeted therapies. The cancer chemotherapy is driven towards personalized medicine and targeted therapy. But if you want to make your targeted drug work, you'll put it in combination with cisplatin. If you want to make your formulation work, you'll put cisplatinum in your formulation. Then it'll work. They're very effective drugs when they work. Like other anti-cancer drugs, they suffer from the same problems. They're not particularly effective in solid tumors, and patients develop resistance quite quickly to the drugs, especially carboplatin. So from the 1990s on, and by the way, I should say this timeline of about 40 years is actually about the same as alkylating agents. The first alkylating agent entered the clinic in 1949 after the Second World War. An orally active alkylating agent was approved in 1989. So it's not actually unusual. It's also, well, show you. So in the 1990s, there were a couple of efforts. Satroplatin was an orally active drug, which was supposed to be orally active carboplatin. Picoplatin contained a pyridine instead of an ammonia to change the kinetics. And we developed this trinuclear compound, which I won't go into the detail of how we did that, but instead of having one platinum, we have three platinums, and instead of the DNA binding sites being separated by about 3.4 angstroms here, the green is the chloride, the green is the chloride. These are now separated by about 24 angstroms. And so they give a totally different type of DNA adduct. And that was the reason why we decided to develop the drug. I can't remember what I said here. Okay. The basis was that the resistance to cisplatin and carboplatin at the time was considered to be tolerability of DNA adducts. And so we made a simple suggestion that if you change the nature of the DNA structure, to modify the structure in a different way, then you may overcome the protein recognition of cisplatin, and you may overcome clinical resistance in that way. So I'll give you a brief history of this compound. Unfortunately, none of these three compounds, they all got as far as phase 2 and even phase 3 clinical trials, but it's a huge barrier to overcome, and none of them have made it to the clinic. If this had made it to the clinic, well, I wouldn't be here. I'd be on the beach, right? So, so I'm still working for a living. So, but to give you the idea of the difficulty in drug development, here's a typical timeline people use of your 5,000 to 10,000 compounds that people like us make. 250 might do extensive preclinical trials. 5 will get to humans, and only one of these will actually make it all the way to an FDA-approved drug. Phase 1 is toxicity, phase 2 is single agent activity, and phase 3 is combination chemotherapy. And the prices go up dramatically. You can do a phase 1 relatively small number of patients, phase 2 also, but phase 3 takes a long time and a lot of money. So phase 2 is actually the sort of graveyard of anti-cancer drugs. That's where the majority of drugs falter, including all the platinum drugs, including our own. Okay, so over the years since the discovery of cisplatin and the entry into the clinic in 1978, we have a much better understanding of what happens, and this understanding of the chemistry, which we had to develop, the chemistry of platinum in cells, is also matched by the advances in cancer biology and cell biology. So the 4 ways a chemist could think of modifying a compound will be drug administration. The agents are given at the moment as IV injections. Strategies allowing better tumor delivery may include oral administration like satraplatin. There are efforts for photoactivity, photoactivation of compounds, and of course there's a large interest in formulations, polymer formulations, nano formulations. Some of these have made it to clinical trials, but none of them have ever been approved, which is actually fairly common for formulations of active drugs, actually. Then we come into metabolism. Cisplatin is a small molecule. It's a soft acid. I hope that at least those of you attending my course will know what a soft acid is by now. And it binds to sulfur. We believe that we need to get active equator species, interaction with thiol-containing proteins such as human serum albumin here with the sulfur, with the cysteine and the methionine, or a peptide such as glutathione will bind to the platinum and labelize the amine ligands, and so the compound is deactivated. So once it gets past some form of metabolism, it has to enter cells, and probably the reason why the compounds are so effective is there are many ways that these small molecules get into cells. They're small, they're diffusible, and there are many mechanisms, both active and passive mediated pathways. Decreased accumulation over the years now has actually been seen now to be a very common cause of clinical resistance. This is something that's come up in the last 10 years. Once it gets past the cell wall and into the cell, we believe that the target is DNA, and the primary cellular consequence of DNA damage and processing are transcriptional arrest and evocation of the apoptotic signaling pathways. Apoptosis in cancer is dysregulated, so in fact the function of any anti-cancer drug is to induce apoptosis. DNA repair, as I say, and enhanced tolerance of adducts also contribute significantly to development of resistance. From here to here, less than 5% of the compound actually makes it to DNA. The rest is either excreted or metabolized. Okay, so as I said, we started this project, as Rose said, it is about 30 years ago. It seems an awful long time. But we had this basic idea that we wanted to extend the relevant platinum chemistry beyond the cis geometry. All of these, there were 30 drugs which entered phase 1 clinical trials based on this cis structure. 3 of them were approved, which is one in about 10%, which, as I say, is actually about average, but all based on this structure. So we know that cisplatin binds to DNA. The DNA adducts are recognized by proteins. These are processed in different ways. And so we suggested that, well, maybe a way of complementing cisplatin therapy and overcoming cisplatin resistance by DNA repair was to produce compounds which will bind to DNA in different manners and thereby not be recognized by the same subset of proteins recognized by cisplatin, and in this way induce some differential tumor effects further downstream. I'm just going to talk about these compounds, polynuclear compounds. We've also looked at some transplatinum compounds, which is developed into a project for antiviral agents, but I probably won't have time to talk about that today. Okay, so I don't have the bending of cisplatin, but you will see that these are models of DNA binding of our long-range cross-linking agents. Oops. What do I do here? Should I escape? Okay. Remember, we now have a long-range cross-linking agent with one platinum chloride here. The central platinum can be displaced by a polyamine, but it only adds hydrogen bonding and electrostatics. And then we have the second platinum chloride bond. So we can form 1,4 interstrand crosslinks, 1,5 crosslinks, 1,6 crosslinks, and we cover therefore a much wider range of DNA than cisplatin does. They're also rather flexible adducts, and we have proven these by NMR, by isolation of individual pieces of DNA, by molecular biology techniques. And what we know is the conformations, and we know that these subset of adducts are not recognized by high mobility group proteins which recognize cisplatin, and so that part of the logic was correct. Because they're long range, they tend to be delocalized. So in fact, the damage to DNA extends even beyond these binding sites, because we see more left-handed DNA rather than bending, as in the case of cisplatinum. And so they're very delocalized lesions, which are more difficult to repair. So that part of the hypothesis proved correct. The compound turned out to be very active, nanomolar cytotoxic. And here is the NCI 60 cell line panel, and what National Cancer Institute has done. For those of you who don't know this is from the COMPARE program, and it's publicly available. You go to NCI website, and you can download profiles of any drug you like. And they take 60 human cell lines. They've actually phenotyped all of these. They know exactly what's mutated and what's present in all of these. And they define a GI50, which is the average of the GI50, growth index 50, of all the cell lines. And then they can plot individual cell lines, whether they're more sensitive or less sensitive compared to the mean. And they can subtract one drug from the other. So in this case, this is a subtraction of BBR-3464 for our drug versus cisplatin. And what this basically says is that bars to the right are more sensitive to our compound, bars to the left are more sensitive to cisplatin. So you see that in colon cancer, almost all colon cancers are more sensitive to BBR-3464. CNS cancers are more sensitive to cisplatin. Ovarian cancers, which is not a surprise because we know that cisplatin is very good in ovarian cancer. But we see renal cancers and some breast cancers are more sensitive to our drug. So this is preclinical. And what was interesting is that we were, of course, this time we had drug company support, obviously, and licensing. In the phase 1 clinical trials and the phase 2 clinical trials, we actually saw activity in these 3 lines, which corresponded to some of the indications in the preclinical studies. Our recommended dose was 1.1 milligram per meter squared, which is about 20 times less than cisplatin. And we saw responses in phase 1 colorectal, pancreatic cancer, and breast cancer in combination with 5-FU. We did not see any nephrotoxicity, so the drug looked very good. In phase 2, then we did phase 2 with patients resistant to cisplatin. So this was more difficult because these are patients who have now been treated with cisplatin for ovarian cancer, and the cancer returns after 6 months, and they come in rather sick. We saw some responses, 5 out of 28, which is actually quite good, and some of these were long-lived. We saw one person who actually survived over 3 years before the cancer returned. These were done in England and outside London. And we saw one response in cisplatin refractory ovarian cancer with a P53 mutation. This is more difficult because this means that the patient has been diagnosed with ovarian cancer but is already not responding to cisplatin or carboplatin, so the cancer is much more advanced. So it was reasonably good. We wanted maybe 25%. We got slightly less than that. But for various reasons, including drug company takeovers and changes, we stopped in phase 2, and partly because we found that at the phase 2 stage in human plasma, the drug was decomposed 3 times, 4 times faster than in mouse plasma, which was sort of a surprise to us. And the trinuclear compound breaks up into dinuclear and mononuclear species by sulfur binding, so you can see very easily if you bind sulfur to this chloride, you have a strong trans influence. You will labelize the bond trans to the sulfur, platinum, nitrogen, and you will end up with this mononuclear species. These are inactive. And these are isolated from blood metabolites, and you can find these if you take this BBR-3464 and react it with glutathione or cysteine in a test tube, you get the same result.